Hello, I'm Dr. Michael Fossel. I'd like to welcome you to the conference. We're going to talk about, for about 45 minutes, something that is rather unusual. We're going to talk about the possibility of curing the age-related dementias, specifically Alzheimer's, but not Alzheimer's alone. The talk will be divided sort of into four sections, pretty much. First, we're going to talk a little bit about what I call assumptions of understanding, where we think we know what we're talking about and frequently don't. Secondly, we're going to talk about unified systems models that really can explain the fundamentals of age-related dementias. Third, we're going to talk about an understanding of the aging process itself at the cellular and genetic levels. And finally, we're going to talk about an optimal point of intervention, which we're going to take to human trials. I got interested in all of this about 45 years ago, 50 years ago, when I was at Stanford here doing a PhD and an MD in neurobiology, and I was teaching in the med school. But what I found is that while all of us are impressed with the development of the nervous system, which is what I started working on, most people, when they discuss age-related disease, tend to sort of shrug and say, well, it just happens. And I find that sort of blasé attitude to be intellectually dishonest. It usually means there's more complexity involved than people are willing to give it credit for. So I got curious about what's really going on. One of the people who played a real role in all this was Dr. Leonard Hayflick, a friend of mine, who made this comment to me while he and I were having a little conversation about a year ago. And he was correct. I think all of us would agree that the fundamental risk factor for age-related diseases like Alzheimer's is aging. And yet, even though we say that, we then entirely ignore the aging process and concentrate pretty much on the histology, the chemistry, the pathology, and so on. But we ignore the role that aging plays. When I talk about the assumption of understanding, this is something that's certainly true with regard to aging and Alzheimer's, but here's an example from early in my career. In the early 1980s, when I started practicing medicine, the assumption was that gastric ulcers, pyloric ulcers were caused by acid and stress, and we treated them that way. Now, it wasn't until 1983 that Robin Warren and Barry Marshall went on to show that the real underlying fundamental etiology was this bacteria you see on the right, which is Helicobacter pylori. And for that, they won the Nobel Prize in 2005, and deservedly so. Now, in 1980, when we were talking about acid and stress as being the cause of ulcers, it was not that physicians were stupid, ignorant, unaware of the sort of data out there, but they simply started with the assumption that they already knew what the disease was about. And that prevented them from digging deeper and asking what was really going on. And I think that's part of what we do so often in science and in medicine. There's an example from 1895 where Lord Kelvin, one of the world's leading physicists of that day, said that heavier than air flying machines were impossible. And he was proven wrong, obviously, some 8 years later, although it took about another 3 to 5 years before the world realized that. But again, it was not that Lord Kelvin was thoughtless or ignorant. No, to the contrary. Very intelligent person, very much aware of the science on air pressure and weight, but a little weak on aerodynamics. And he had that same assumption of understanding. He thought he understood the basic assumptions, the basic premises of heavier than air flying machines, and he was simply wrong. But we do the same thing in medicine as well. We often forget about underlying causes and we look only at the superficial aspects of it. So here's an example from microbiology. If any of you know anything about plague, it usually comes in 3 different clinical syndromes. There's bubonic plague, which most of the general public is aware of, pneumonic plague, which some people are aware of, and septicemic plague. And if you knew nothing at all about microbes and all you knew was the clinical presentation of these diseases, you might assume that they are not related. Well, they are. They're all caused by Yersinia pestis, and it depends very much on the point of entry in the body and the immune response. So they are really one disease, but at the surface level, in terms of the clinical presentation, they look different. Much the same is going on with regard to age-related dementias. So whether we're looking at Alzheimer's, Parkinson's, frontotemporal dementia, vascular dementia, or any number of other dementias, which we'll come back to, they are probably sharing an underlying fundamental cause. We begin to suspect this when we see things like the overlap involved that's suggested here. For example, Alzheimer's patients frequently have motor problems. Patients with Parkinson's disease frequently have cognitive problems. And likewise, when we take any of these patients to autopsy, we frequently see vascular etiologies as well. But the same is true when we expand the alphabet in a sense. Look at things like Lewy body dementia and posterior cerebral atrophy and so on. What you find is that the reality is that while these clinical syndromes are real. There are also atypical dementias where people don't quite fit into any of these categories very neatly. And there are mixed dementias where people fit into a category, but they have certain presentations that are more typical of another category. And the result has been that over the past decade or so, you begin to see professional interest groups and, in fact, global conferences on atypical dementias and mixed dementias and so forth. And there has been a growing feeling, informally perhaps, that there are shared fundamental etiologies that underlie all of these age-related dementias. And they're often typically called age-related neurodegenerative diseases. Now, it's not clear what that fundamental underlying etiology is, but that's begun to become something of a global consensus. About 2 years ago, the Alzheimer's Association invited me to help organize a conference in Washington on animal models of Alzheimer's. Now, as many of you are aware of, this is a statement that's very typical. Everything works in mice and nothing works in humans. Of course, the truth is not everything works in mice. But the problem is many of the basic research projects we do on the dementias work very well or fairly well in mice, but they don't translate well to human trials and we don't get an effective drug. And the question is why? Now, there are any number of reasons that are involved here. I pointed out that part of the problem is that there are 25% of protein expressing genes that we do not share with mice, and that many of the mechanisms that we delve into are related to mechanisms that are sort of native to those non-shared genes. So it's small wonder they don't translate. Now, there are a number of fundamental mechanisms we do share, as we'll see. But the other problem I pointed out was that we lack a single unified systems model to explain how these diseases work. And we'll come back to what I mean by that model. But after I gave that talk, I was invited to write this paper by the person who turned out to be the editor-in-chief of Alzheimer's and Dementia, the world's leading journal on dementias. He then recused himself from the editorial process, asked to join our scientific advisory board, which he did because he was impressed by the model. Now, the paper itself went through 8 reviews, very complimentary, and they requested that I write a slightly longer paper, which I did. And the paper came out almost a year ago and promptly generated more than 600 global reprint requests and considerable interest from both investors and from people who wanted me to give talks like this one. But the gist of that paper was not simply that it got a lot of reprint requests, but that it offered an explanation for many of these age-related diseases that was consistent with all the known data. It was predictively valid in terms of big pharma clinical trials, and it offers an innovative point of intervention, which we're going to look at now. So when I say that mice and humans often don't share mechanisms, we're looking at intermediate pathways. Mice and humans share many fundamental pathways, like those of cell senescence, cell aging. But when it comes to, for example, the way the mice handle beta amyloid, it's slightly different physiology. So it's, again, small wonder that things don't translate from one to the other. And this is true almost no matter what species we look at. The same is true when we look within human species and we ask ourselves what's going on with different age-related demential diseases like Parkinson's and Lewy body dementia and posterior cerebral atrophy and so on. What you find is that the genetic and epigenetic backgrounds of individual patients are slightly different, but the fundamental mechanisms are the same. And these, again, involve cell senescence and cell aging, which we'll come back to. So the outcome is a different clinical presentation, a different clinical disease, if you will, but shared fundamental mechanisms expressed slightly different because we're all slightly different people. And I suggested what we needed was a unified model and a systems model. By unified model, I mean that it had to explain the heterogeneity within specific dementias. For example, why do some patients get Alzheimer's at age 45, other patients at age 75, and other patients not at all? Why is it that we see different kinds of dementias? Why that I get Parkinson's, you get Alzheimer's, and our patient gets Lewy body? Why are these dementias different? What makes that happen? Also, the differences between species. Why is it that mice, rhesus monkeys, gorillas, cynomegalops monkeys, all these get slightly different kinds of cognitive decline with age? And also, multiple upstream mechanisms. Why is it that trauma, for example, head trauma, increases your risk of, for example, Alzheimer's or Parkinson's? Exposure to some toxins like paraquat increases your risk of Parkinson's. Why is it that many encephalitides, whether they're viral, bacterial, or otherwise, can increase your risk for age-related dementias? Why does that happen? Why is it expressed downstream in multiple biomarkers like beta amyloid or alpha-synuclein or tau tangles, mitochondrial dysfunction, and so forth? Why exactly do these things occur? So we need a model that's unified and can encompass all of these explanations. And it also needs to be a systems model. By systems model, I mean, it's got to explain how these things interact, not simply the components, but the entire system. Let me give you an engineering example. This is an Airbus 380. In the upper right, you see a Rolls-Royce Pro 15 jet engine. In the lower right, you're seeing a Trent 900 turbofan with a single turbofan blade. And if a turbofan blade fails, you should not be asking what's wrong just with the turbofan, but what's wrong with the turbofan in that Trent 900 turbofan, in the Rolls-Royce Pro 15 jet engine, in the Airbus 380 at 35,000 feet, while operating at hundreds of miles an hour, hundreds of RPM at high temperature. In short, the component is not the issue, it's the system itself. So if we have a failure of a component, it should not be simply a question of the component, but a question of the entire dynamic system that we need to look at. Well, the same is true in medicine. So for example, this is the Ebola River, and these are signs and symptoms of Ebola disease, something that I taught a semester course on. These are signs and symptoms, but they are not the disease. They're simply components of the disease, if you will. They're clinical biomarkers of the disease, but they do not represent the underlying systems process. If we want to be able to understand Ebola, we need to understand that there's a virus involved and an immune response in a very complicated human body. It is not simply the matter of the components, that is, the biomarkers or the signs and symptoms. The same is true, for example, regarding COVID infections. These are typical for COVID infections, but they are not the disease. The disease is a process that involves the point of attack of the COVID virus and what happens with the immune system in various parts of the body. So again, signs and symptoms in medicine are not a disease. They are simply outcomes of the disease. And the same really is true with age-related dementias. If we look at aging itself, these are typical macroscopic biomarkers of age-related disease. Declining glomerular filtration rate, arterial disease, poor healing, sarcopenia, all of these are really common things that we see in our patients or ourselves, but they're not aging. They are outcomes of aging, but they're not the process itself. Biomarkers are not a disease. Biomarkers are not aging. The same is true if we look at the microscopic level. So whether we're looking at declining ratios of ATP to reactive oxygen species, telomere shortening, declining DNA repair, inflammatory changes, whatever, these are biomarkers, but they are not aging. They're not the process. People who concentrate on biomarkers are missing the point, just as people who concentrate on signs and symptoms of a disease are missing the point of understanding the actual process of the disease. The same is true if we look at particular lists. This is a list compiled by an organization that purports to try to understand aging. And they've listed 9 different biomarkers of aging, but none of these are aging. They're outcomes of aging. They're, if you were, symptoms. Symptoms are not a disease. Biomarkers are not the process. If we look at biomarkers for dementia, we see the same thing. Alois Alzheimer originally described Alzheimer's more than 110 years ago and warned us even then against the idea that beta amyloid plaque was the cause. It's an outcome, it's a finding, it's a biomarker, it's a component, but it's not the disease. The same is true of all these other things, whether you're looking at blood-brain barrier failure, lymphatic changes, inflammatory changes. All of these are very prominent in the literature, but they are not Alzheimer's. They are biomarkers of Alzheimer's, but they do not explain the process of the disease, which is what we need. The same is true when we focus on genetics. These are some of the common genes that have been associated with Alzheimer's and some of the other dementias, and we could go on with dozens of others. But genetics are not the disease. Genetics contribute to the disease. They may be risk factors for the disease. They may be correlated with the disease. But ApoE4 alleles, for example, 2 ApoE4 alleles are not Alzheimer's. There's not 100% penetrance, for one thing. So the question is, what's really going on with the underlying process? Not simply the genetics, not simply the risk factors, but the disease itself. There's a common statement in science, which is if you can't measure, it's not science. But we need to go beyond that. It's not enough to measure things, it's enough to understand them. If you cannot understand the process, then you can't actually cure it. It's necessary that we not simply measure biomarkers, alpha-synuclein, beta amyloid, tau tangles, but we understand how those got there and find the most effective point of intervention clinically. So the question then comes down to, do we truly understand the age-relating process that results in age-related dementias? Because only if we truly understand aging can we intervene and cure age-related diseases. I first got involved in this in a public way 24 years ago when I gave the first talk on this ever at the National Institutes of Health. And the question I was addressing was, what really is aging? Do we truly understand the process? Because in many respects, we did not, and maybe do not. The result of that was me putting out the first book on the potential for reversing it, the first articles ever published in medical literature on the potential for reversing age-related diseases at the genetic and serial level, the first and still only textbook on this area with Oxford University Press, Cells, Aging, and Human Disease, and a more recent book for the general public, the Wall Street Journal praised as one of the best 5 science books of the year. But the question that all of these have tried to address is the same, which is, What is the aging process? So let's look at that a little more carefully. These are typical species that age. Human beings, for example, have a mean lifespan now of about 75 years. Gorillas, as in the upper left with Coco, the gorilla spoke sign language, just short of a half a century. I actually was her babysitter for a year. She spoke sign language better than I do, which is humiliating but true. Typical domestic animals, for example, dogs and cats, live about 2 decades, depending on the breed. And typical laboratory rodents live about 2 or 3 years, again, depending on the breed involved. Now, you can't very well wave a wand and say aging is just entropy without explaining why different species have different rates of aging and therefore different rates of entropy, apparently. Now, most people at this point would simply say it's genetic, but genetics are not an answer. You have decided that genetics play a role in it, and that's true, but you have not explained why genetics affect the rate of aging. And the same is true even when we look within the human species. These are 30 progeric children. I was gathering them together from all over the world every year. This is the year I took them for a tour of the White House. And these children die overwhelmingly of age-related diseases. Their average age of death is 12.7 years. They are, as you look at the kids here, all bald. The ones who apparently have hair are wearing wigs. They have thin skin that heals poorly. They have osteoarthritis, osteoporosis. And the most common cause of death is strokes, heart attacks, and aneurysms. They have overwhelmingly age-related arterial disease. Why? We know the linked gene, and that is the lamin A gene. But again, that's not an explanation. It doesn't explain the process that advances aging in these children. It identifies a gene that's a risk factor, but it does not explain the dynamic process. It's a biomarker, not an explanation. The same is true when we look at particular cell groups. For example, every single one of you who've been listening to me here is derived from a single fertilized ovum. And the ovum derived from your mother. It was several decades old then. She got it from her mother. It was several more decades old then. And that line of cells can be traced back in an unbroken lineage for 4.5 billion years. And during that time, the cells changed a great deal, but they showed no evidence whatsoever of aging. And yet there's an inflection point, and many of your somatic cells now show aging changes. The same is true of your mitochondria. Every mitochondria in your body right now derives from your mother. And again, those can be traced back for 1.7 billion years in the eukaryotic cell line, but there was no evidence of aging whatsoever. And yet many of your mitochondria show age-related changes. Now, we'll come back to that. But the question is why? Why is aging not something that is unavoidable? It occurs in certain cells and not in others. Aging is clearly more than entropy. It is entropy versus maintenance. In fact, maybe an analogy you might use is gardening. If I'm looking at a garden, these are two of my gardens, the reason they do well is not because there are no weeds, it's because there's a great deal of weeding, that is maintenance. So entropy is very typical. What distinguishes a garden that sort of goes downhill from a garden that does very well and shows no aging changes is a great deal of maintenance or weeding. The same is true if we look at what's called red queen theory. And this applies even within cells. This is a quote from Alice in Wonderland. It takes all the running you could do to keep in the same place. And the same really is true of cells. For example, a germ cell lineage to be able to maintain itself for 4.5 billion years without aging means that it needs to take all the maintenance it can to keep up with entropic changes. And that's true within our cells. So as we age, what you find is the germ cell line manages to maintain itself flawlessly for billions of years, but typical somatic cells do not. This is an 80-year-old with a hand in the lower left there you're looking at. And if I look at the keratinocytes, the epithelial cells, the endothelial cells, the macrophages, any of the cells in that hand, what you find is their ability to maintain themselves has gone downhill. That is, they're no longer maintaining themselves the same level of efficiency they did when this was a young person. Well, if you consider young cells versus old cells, what you find is the difference is the pattern of gene expression and, for that matter, telomere length. It is not a matter of different genes. So the difference between young cells and old cells now is not that the genes have changed, is that the pattern of gene expressions have changed. So the difference between me at age, for example, 7 and now me at the age 70 is not a difference in my genes, largely. It is a difference in the pattern of gene expression. And this is associated with the shorter telomeres. Now, telomeres do not act as the overwhelming cause of aging by any means, but they act as a conductor, maybe a moderator for the entire orchestra of gene expression. You might say the difference between the tune the young cells are playing and the old cells are playing is set by the conductor. And the conductor chooses the score and sets the tempo, but it is not that the orchestra has now, the violin is broken or the piano is out of tune or the oboe player has a split reed. The difference is the tune that they're playing. And the tempo turns out to be important because in young cells, no matter what molecular pool you're looking at, for example, beta amyloid, tau tangles, or for that matter, collagen and elastin in the skin, no matter what molecular pool you're looking at, you find that in young cells, there's a very rapid rate of turnover, a recycling rate, if you will. And the outcome is there's very little accumulation of damage in those molecules. But as you get older, your telomeres shorten, the pattern of gene expression changes, you find turnover slows, and there's a gradual increase in the number of dysfunctional molecules, the result of which is dysfunctional cell. For example, again, beta amyloid or tau tangles. You can even look at it mathematically. This is an example taken from my textbook. And what you see is that in young cells, you have a very rapid rate of turnover and a very low rate of damage. Whereas in older cells, as turnover slows, the damage that's accumulated goes up over time. So what's going on is, no matter where we look in cells, as the recycling rate goes down, the equilibrium damage over time goes up. And in consequence, so does age-related disease. For example, again, the age-related dementias. Now, we can look at this within the brain. So let's look at typical examples of, say, Alzheimer's disease. And in this case, we'll focus on beta amyloid, although we could look at other molecules as well. What you find is that in young cells, the production, secretion, binding, reuptake, and degradation rates, which is the recycling of this compound, are all fairly rapid. But as cells get older, you find that the rate of turnover goes down, and the result is you begin to get some beta amyloid microaggregates. And you might ask, what's the difference between different genes? For example, if you're lucky and have two ApoE2 alleles, you tend to make beta amyloid molecules that have very little stickiness to them. They don't tend to form microaggregates. And so even though as we get older, the rate of recycling goes down, you don't get many residual microaggregates. But if you have two ApoE2 alleles, you tend to make stickier molecules. And in consequence, as you go through the ages of 40 and 50 and 60, you have a much more rapid accrual of beta amyloid microaggregates and hence of disease. But regardless, as you get old enough, the rate goes down so far that you begin to get formation of beta amyloid plaques. And the result of that is synaptic loss, dysfunction, dead neurons, and frankly, clinical dementia. The same thing is true no matter what we're looking at. We can look at beta amyloid, tau tangles, or other things as well. So let me give you an example of this. In this case, what I'm going to do is share with you a particular video that makes this point, I think, very nicely. Here it is. All right, take a look at this.
Alzheimer's disease is one of the most common age-related neurologic diseases. It involves degenerative changes and cell death in the cortex of the brain, with gradual loss of cognitive function. Many factors contribute to the disease, but the fundamental process is age-related epigenetic changes in the glial cells of the brain. Neurons depend on the glial cells to function normally. Microglia, one type of glial cell, constantly produce and absorb molecules in a continual recycling process. One common example, beta amyloid proteins, are produced and secreted by the microglia. Some of these proteins are sticky and apt to form clumps. But they are usually reabsorbed back and broken down. This molecular turnover is rapid in young cells, but as the cells age, molecular turnover slows down, leading to a gradual accumulation of the microaggregates around the cells. As molecular turnover slows down even further, the microaggregates grow larger, forming beta amyloid plaques and damaging the neurons. Over the years, these plaques increase, as does the damage to the neurons, which begin to fail and die. Molecular turnover slows down because of subtle but pervasive changes in gene expression in aging cells, which are the result of telomere shortening. Telomeres are the regions at the end of each of our chromosomes that not only protect the genes, but also serve as aging clocks, shortening over time. Over the human lifetime, cells may divide many times, and each time, the cell replicates its chromosomes. The replication protein complex attaches to the end of the DNA chain, but can't read the tiny segment of telomere that lies beneath its attached area. So, with each replication cycle, the telomere grows slightly shorter. And as the telomere shortens, it affects the rate and pattern of gene expression throughout the entire chromosome. As our cells divide, telomeres shorten, gene expression changes, protein turnover slows, and cells fail and die. The final result is Alzheimer's disease.
So let me take you back now to the slides and continue on and tell you a little bit more about what is going on. We've addressed a little bit about the beta amyloid, tau tangles, issues like that, but the same is true within the rest of the cell as well. For example, if we look at how the mitochondria function, you find that the bulk of the aerobic enzymes, 99% of them actually, derive from the nucleus. And as telomeres shorten in the nucleus, you find that there's the same slowing of recycling of the aerobic enzymes. The result is, again, you have more defective aerobic enzymes, the mitochondria, hence you have more in the way of free radicals. And you also have leakier membranes. The lipid membranes that surround the mitochondria are also being turned over, and the rate of turnover goes down, so the membranes begin to oxidize and have more leakage. Furthermore, the scavenging molecules that generally are responsible for grabbing onto and binding the reactive oxygen species, such as catalase and superoxide dismutase, are also being recycled more slowly. So you get poor scavenging, and the molecules that are damaged also get turned over more slowly. So for 4 separate reasons, you begin to find that you have more and more damaged molecules in cells as we begin to get aging in cells. And this is also true when we look at DNA damage. The DNA molecules are the only molecules in your body that you actually repair rather than simply recycle. But the problem is that the families of enzymes that are responsible for DNA damage also are slowed down. They're less apt to be recycled. So detection is slower, multiplied by excision being slower, multiplied by replacement being slower, multiplied by ligation being slower. And the outcome of that is that you find that the incidence of malignancy goes up. So this is true whether you're a 2-year-old mouse or a 100-year-old human. The increase in malignancy with age is not simply a result of exposure over the lifetime to carcinogenic substances. It is also a result, largely, of the decrease in DNA repair because your cells are no longer capable as they were when you were younger. Well, what is going on then is that telomeres modulate that pattern of shortening. The telomeres do not cause aging. Let me stress that. Telomeres no more cause aging than genes cause evolution. Genes clearly play an important role in evolution, but it would be a misconstrual to say that genes cause evolution. They're a key player, but they're not the cause of it. In fact, cause is probably the wrong verb in many ways. The same thing is true here. Telomeres by no means cause aging. They're simply a central player in aging and cell aging and ultimately in age-related disease. Furthermore, telomere length doesn't matter. It is not a matter of length, it's a matter of change in length. So for example, there are mice that at birth have telomeres 10 times longer than mine, but lifespans that are 48 times shorter than mine. Because the question is the rate of telomere loss, and more importantly, the change in the patterns of gene expression, the change in turnover of molecules, and the effect that has on cell function. So for example, it turns out that while telomere length has no ability to predict really lifespan between species, it does very nicely if you look at the rate of telomere loss in predicting lifespan between species. The same thing is really true in human beings too. It's not a matter of your telomere length, it's a matter of how fast you're losing it, particularly in special cell populations that are responsible for disease. This is a complicated diagram taken from my paper that you're welcome to get a copy of. Really what this says though is that upstream there are any number of things that affect the rate of cell division and therefore the rate of cell aging. And downstream there are any number of biomarkers that are a result of these changes in cell aging. So for example, we know that exposure to head trauma increases your risk of cell aging. For example, in this case, ultimately beta amyloid plaques with beta amyloid. Same is true of exposure to toxins. As I say, exposure to paraquat can increase your risk of alpha-synuclein aggregates ultimately, because you're again increasing the rate of cell aging. Same is true of radiation, skin, for example, but bacterial infections as well as bacterial, chlamydial, fungal infections, viral infections, even prion diseases can all be affected here, as can vascular dementias, for example, hyperglycemia or hypertension are risk factors. And all of these sort of feed through cell division, shorter telomeres, altered patterns of gene expression, and cell dysfunction, resulting in the dozens of things we see downstream. Now, let me say that in some sense, none of this matters. The question for me is not what's causing this, but where's the single most effective point of intervention, both clinically and for that matter, financially? And the answer is that the upstream targets tend to be inefficient. If I, for example, make sure you never have any head trauma, you still can develop Alzheimer's. If I can absolutely normalize your blood pressure and your blood glucose, you still can have vascular disease. If I can absolutely prevent you from having exposure to radiation, your skin cells and other cells still show age-related changes. So these are worthwhile targets, but they're not the most efficient targets. The same thing is true downstream. For example, there have been more than 1,100 interventional trials for Alzheimer's disease, and by global consensus, none of these have resulted in a change in the course of the disease progress. So, for example, beta amyloid appears to be not the most efficient target. Not to say it's not a worthwhile target to try for, but it's not an efficient target. The most efficient target clearly turns out to be cell aging. And the reason I say this is because it works very well, actually. For example, the first time that human cells were ever had their age reversed was now 23 years ago in this classic Bodnar paper, showing that you could reset the pattern of gene expression. The first time this was ever done in human skin cells was done 20 years ago, and the same is true with regard to bone cells, some 19 years ago, or for that matter, vascular cells 19 years ago. Here we're looking at vascular endothelial cells. So we clearly know that we can reverse aging in human cells in the laboratory and in human tissues in the laboratory. The question is, can we do this with organisms? And the first time that was ever done was actually 2011, followed by this article in 2012, using a technique that we can now apply to human clinical trials. And what it shows is that if you reset telomere length in active animals, old animals, you can actually not only reset telomere length and reset cell aging, but what you do is reverse the cognitive decline that we see typically in these animals. So in consequence, we're going to take this to human clinical trials. This is our scientific advisory board. We have, for example, people like Zavin Khachaturian, the editor-in-chief of Alzheimer's Dementia Worldwide, Richard Mose, who is the chief scientist for the Global Alzheimer's Platform Foundation, Mamoun Azouz, who leads the Translational Genetics Research Institute in the UK, Suzanne Hendricks, who's known worldwide for designing Alzheimer's trial and the statistical analysis for them, for example, the biogenescience studies, Joe Araujo. If anybody does large animal work with Alzheimer's, they probably talk to Joe, or Kurt Whittemore, who's now a postdoc at Harvard, who did some of the original work with Miriam Blasco in Madrid at CNIO. We also have an excellent clinical advisory board. These are professors of neurology known worldwide for being, let's say, conservative and skeptical. And we brought them on because we wanted to make sure we do this right. So we've got Maria Marcellus from Sunnybrook in Toronto, Lon Schneider, the Keck School of Medicine at USC, Steve Arnold at Harvard Medical School, Russ Swerdlow at KUMC, who also is the director of the Alzheimer's Center. All of these people have been doing Alzheimer's research, human clinical trials for a number of years, and they've signed on to make sure that we do this right. In short, we're going to take this to human clinical trials. We're going to be using a single dose gene therapy to reset the pattern of gene expression using an AAV9 virus, a human telomerase gene, and we're going to be targeting glial cells and neurons in the brain. We have had already informal discussions with the FDA and CBER, and we intend to take this to human clinical trials soon. Now, note that what we're doing is not killing off senescent cells, but restoring senescent cells to their normal function as they were prior to the onset of senescence. And we have good reason to think that if we do this, we can do something that has never been done before, which is not simply to slow or stop age-related dementias, but actually reverse some of the cognitive decline we see in these diseases. Should you have further questions or wish to talk to me further, please get in touch with me. I'd be delighted to hear from you. In the meantime, I hope you have a good conference, and I wish all of you a happy holidays. Thank you very much for listening to me, and good luck to you all.